So today we're privileged to have visiting fellow Professor Tracy Bale here with us in Melbourne. Tracy will deliver her talk, Stressed Parents Shaping the Brain Before, During and After Birth. The lecture will run for around 45 minutes or so, followed by a brief question time. So to introduce Tracy, Professor Tracy Bale is Professor of Pharmacology and Director of the Centre for Epigenetic Research in Child Health and Brain Development in the School of Medicine at the University of Maryland, Baltimore. She completed her PhD at the University of Washington in the Department of Pharmacology and her postdoctoral work at the Salk Institute with Wiley Vale, who's really one of the legends of neuroscience who sadly passed away. Tracy was Professor of Neuroscience at the University of Pennsylvania for 15 years, prior to her move to University of Maryland, Baltimore. Her research focuses on understanding the role of stress dysregulation in neurodevelopment and psychiatric disorders, and sex differences that underlie diseases, their vulnerability, and also using models such as mice to understand these disorders. She's interested in developing models of parental stress and germ cells and their involvement in transgenerational epigenetic programming of neural development. Tracy serves on many internal and external advisory committees, panels and boards, has served as chair of major committees in North America, reviewing editor of the Journal of Neuroscience, which is a leading journal in our field, has been recipient of many awards in this area, and has won prestigious awards from the Society for Neuroscience, the largest society for neuroscience research in the world, the Richard E. Weitzman Memorial Award as an exceptionally promising young investigator of the Endocrine Society, the Medtronic Award from the Society for Women's Health Research for outstanding research that has led to the improvement of women's health, and recently the Daniel H. Efron Award from the American College of Neuropsychopharmacology. Tracy was recently elected president of the International Brain Research Organization, one of the great organizations of neuroscientists around the world, one of whose remits is to support neuroscience around the world, including in developing countries. So it's my great pleasure to introduce Tracy to deliver her lecture.
I want to first of all say an incredible thank you to Tony for organizing this trip for us. It's incredibly exciting to be here in this beautiful city. I have visited Australia a number of times. This is my first trip to Melbourne, and it's a beautiful city. I'm so wowed by the generosity of the people here. We were wandering around last night, and the beautiful architecture, the incredible food. It's just a beautiful, beautiful city, so we're so happy to be here. And I want to say that I'm also really excited because it's not every day that as a scientist you get an opportunity to talk about your research to the public in a way that is both more engaging and to really discuss the importance of the things that we work on and sweat and tears we put our lives into to really try to understand the brain, and especially the research that I do, and I know that Tony is interested as well, is the developing brain especially. And I hope that tonight, through a little bit of levity, because it is summer and it's late and everybody needs a little levity in their lives, and the world is a very stressful place. My lab studies stress, so I know what I'm talking about. And so in order to understand a lot of the science, I think that we need to put it into a context that is both engaging and interesting. And so I hope that you travel this journey in the next 45-ish minutes with me, and that at the end we'll have plenty of time for questions, which I always find to be the best part of getting to engage with the public, and is to figure out what you're concerned about and what you're interested in, because it shapes a lot of the research that we do around the world. So with that, and that idea of it is evening and keeping everybody awake and on their toes, we're going to start off with a little bit of comedy. So I was always told that you should always start off a talk with something funny to get everybody loosened up a little bit, because science can be tough. So two scientists and a politician, at least a politician of sorts, we'll come to that, walk into a bar. And they happen to notice two alcoholic mice, a mother and her son, and they're sitting on bar stools, madly lapping gin from thimbles. The mother mouse looks up and sees the three, and she says, "Hey, geniuses, tell me how it is that my son is in this sorry state." The first genius, Gregor Mendel, says, "Clearly it's your bad genes have led to this event." Freud says, no, it was your helicopter parenting that has led your child down this horrible path. This is big in the US. I don't know if you have helicopter parents here. We have a lot of them in the US. Of course, Trump just jumps right on in there with clearly your ancestors were losers. I apologize for Trump, by the way. So as the three continue to argue, in walks 5 very smartly dressed men from an 80s pop band you may recognize, who settle the debate with one word: clearly. You need more Vegemite. You can't not dance to this song. If you were not alive in the 80s, dance to this song. He just smiled and gave me a Vegemite sandwich. Surely, if every child just had a Vegemite sandwich and a smile, we might get past that. So in thinking about tonight's talk and thinking about the research that my lab does, and a lot of the research that's going on here at the Florey, is really thinking of the consideration of your genes, right? Everybody in this room has a mom and a dad that you inherited your DNA from. And for what the last 100 or more years has told us is that we are everything about our DNA, and this is true. It is exceptionally difficult to outrun your DNA. But that doesn't mean that the environment cannot have an influence, and that is the piece of the puzzle that we're talking about tonight. So our genes, are they or are they not our destiny? And I would argue they may not be our destiny, but surely we need to take into consideration those things that we are at risk for. And I will also highlight the fact that we are all sitting here tonight means that our ancestors had the DNA which gave them resilience and survived. And if they hadn't survived, whether it was everything from Viking invasion, I'm saying that because I'm Danish, or anything from environmental famines, etc., we ourselves would not be here today. So yes, we may be our DNA, but the environment has had an influence as well. And as neuroscientists, we like to refer to this as the G by E by D. And this is just an easy way of saying that we are a composition of our genetics, by our environmental experiences, by those developmental windows that those experiences happened. So your DNA are your risk for cardiovascular disease that you inherited from mom and dad, or let's say for schizophrenia for that matter, and is influenced by the environment that you were raised in. It could be the environment pre-conception, the environment while you were gestating, the environment postnatally, the environment of puberty, the environment across maturation in the lifespan has a big influence on our brain. And those are the developmental windows when they happened. So it's the genetic composition by the influences of the environment, and that environment is very broad. It could be nutritional environment, stress environment, trauma, but it can also be enrichment. And that is something that the neuroscience world is incredibly interested in trying to define, because we are not just dogmatically predetermined toward disease. We can also be incredibly resilient. And again, the D is those developmental windows. Was mom stressed prior to getting pregnant? Was dad stressed prior to getting pregnant? Was your mom exposed to an infection during her pregnancy with you? Prior to your child launching into puberty, was there a particular traumatic event that happened? All of these factors determine much about how our brains develop and mature, and determine about how we interact with our environment. So in thinking this through and thinking about how our genetics interact with our environment, we can go back to Gregor Mendel. So we all know that Gregor Mendel was an incredible geneticist who was studying the importance of the influence of the environment. Okay, hold on. Oh, there we go. That was really critical that we have the P that falls on Gregor Mendel's head. So in order to understand our genetics, Gregor Mendel, spent a lot of time as a monk in his pea patch trying to breed different types of peas, and we learned a lot about genetics. Everyone remember from school your Punnett squares and looking at the wrinkled and smooth peas. These are incredibly important, and we are composed of our DNA. But what Mendel didn't really have time in his years out in the pea patch was to think about how the environment, if there was a drought one year or lots of rain one year, did that influence the epigenetic marks, I'll come back to that, of those seeds that gave rise to next year's crop? And that is something that is missing in Gregor Mendel's understanding of genetics. So this sounds a little bit like evolution, right? We're thinking back to evolutionary perspectives. Unfortunately, evolutionary perspectives are very, very slow, and that the adaptation, I don't understand what happened. There we go. Oh, there we go. It's glacially slow. That we know that in fact the environment which is influencing how in fact that we adapt to our environment sounds a lot about like evolution, but evolution takes a long time. It doesn't really account for how your effects of your environment might be passed on and affect the development of your offspring, right? That's not evolutionarily, that's too fast. So when we look at these together, both Mendel and Darwin, and think about how they may accommodate the effects of the environment, the opioid crisis, stress, infection, or our current obesity epidemic, right? We have not evolutionarily kept up pace in order to adapt our brains and our responses to a very heavily food and drug available environment in terms of our risk for disease or resilience. So then some people will say, this sounds a little Lamarckian, doesn't it? So we all remember that Lamarck's idea was that if a giraffe had a long neck and was able to reach the most delicious leaves, that this effect could be passed on during gestation to her developing offspring and therefore have a longer neck itself, giving advantage to that next generation. Now, Lamarck was thought to be incredibly crazy, and I'm not suggesting he wasn't, but there is some truth to the way that he conceptualized the effects of the environment on our DNA. And that is really what we're terming as epigenetics. We are not changing our DNA. What we're changing are the modifications on our DNA. And those modifications can determine a lot about how we grow and develop, and if they are in our germ cells, how the next generation or future generations as well. So this is what epigenetics are. If you look deep within the chromatin, focus way, way in, you can see that we have histones and around a nucleosome, and those modifications, as well as methylation around our DNA itself, and the production of non-coding RNAs really comprise what we know to be epigenetics. So these are really just mostly chemical modifications within our DNA that are placed by the environment. These are cell type specific, tissue specific, and they can be temporary or lasting. So the idea that the environment can influence how any given gene or many different genes can be expressed and when they're expressed is really what we think about in terms of epigenetics. Understanding how the environment places these marks and how they might be lasting is really on the cutting edge of this field to try and understand those traits. So what are stresses in our environment? Well, in the US, there was studies that were done decades ago, done by Kaiser Permanente, which is a health insurance company. And these studies were initially done in California, so a largely middle class, sort of middle and upper class white population. And the idea was they wanted to understand if you were exposed to adverse events, different traumatic events prior to really the onset of puberty. So all the point during your brain is maturing and developing, exposed to the environment prior to being an adult. Did it give rise to risk? Remember, this is an insurance company. Give rise to risk for disease. And if they could determine what those the most likely adverse childhood events were, they might be able to predict who was at risk. Again, insurance company. So what they determined from these studies was, in fact, that here's the ACEs listed here. These are 10 adverse childhood events, and they include things such as death of a family member, were you molested or raped, did you have a parent who was incarcerated? So pretty traumatic events, although one of them is divorce. Pretty traumatic events. Of these 10, they noted the cut-off point, or the sweet spot here and for predicting disease, was if you had been exposed to 4 or more of these prior to puberty, you were at a significant risk throughout the remaining part of your lifespan to present with some kind of disease or disorder, again, depending on your genetic DNA background. And this is just showing you all of the things that would increase and possible risk outcomes for these exposures to these adverse childhood events. So again, 4 or more being really the point at which you were crossing over that line for being at risk for either behavioral changes in terms of addiction, et cetera, or other diseases. I'm particularly interested in brain development, so of course I'm particularly interested in women who are pregnant. So if you look at adverse childhood events, so those exposed, women who are now currently pregnant, but themselves were exposed to adverse childhood events, and you take a survey across the United States of America, you can see women who are currently pregnant and were exposed to 0, 1, 2, 3, or 4 or more in yellow here, is about 12.5% at that point at which you were thinking them as being particularly at risk themselves, and they are now pregnant, and you can imagine that may impart an effect on their future offspring. I live in the city of Baltimore, and I'm particularly interested in stress and traumatic events that happen in the population in the city in which I live, because we do a lot of translational research in my lab. So as Tony pointed out, I do a lot of things using mouse models where I can manipulate the environment, but we also do in every one of our projects in the lab a translational approach where we work with humans as well to understand the things that we're finding in the lab. So I wanted to understand, in a study that was done in the city of Baltimore, how many traumatic events, and I will tell you for those who don't know, Baltimore, if anyone's seen The Wire, okay, don't watch it. So the city of Baltimore is a largely African-American, is majority African-American. It's somewhere around 60% African-American, and there's a history of racial discrimination and disparities in the city. And so there's a lot of trauma and a lot of violence that have happened over the last many decades. And so we're particularly interested, especially in women and their mental health, and the impact it has on their children as well. So doing this exact same survey in the city of Baltimore, you get a very different result. 61% of pregnant women within the city, again, attending to their pregnancy and health needs at the University of Maryland, so a public institution, 61% have experienced 4 or more traumatic events prior to the onset of puberty. And that's a lot. And I think that's incredibly important as we move forward in trying to understand why is that. Because in the United States of America, we have a huge problem with discrimination, stress, and health disparities. In the United States of America, a woman who is African American or Black is 4 times more likely to die during pregnancy or postpartum, as is her child, regardless of her socioeconomic status. So an African American woman with an advanced degree, a PhD, an MD, a law degree, is still 4 times more likely to die than a Caucasian woman with an 8th grade education. This is not about socioeconomic access. This is actually something very specific to racial discrimination and the constant and persistent stress that these individuals encounter. And that is something that we're very interested in studying and modeling in the lab. So what about Australia? So I looked up some statistics, and as Tony pointed out, and the research going on here at the Florey is also very interested in understanding mental health issues. So somewhere around 25% of adults in Australia, or 5 million people every year, report having experienced significant number of adverse childhood events during, again, their childhood. And we know just ongoing right now, the experiences of natural disasters such as the bushfires, the wildfires that are going on all over Australia, recent flooding, there's flooding again up in Queensland, there's been history, lots of individuals, I know there are scientists both within the US and Canada who do all sorts of studies here in Australia based on Queensland floods and those traumatic experiences, shootings and other violences as well. And in fact, if you look at gun deaths in the US and Australia, of course you have stricter gun laws than we do in the US. You can see that, again, looking at the gun deaths because of those stricter gun laws, but not 0, of course. And so those are all traumatic events, depending on what city you live in, the experiences that you have. Ultimately, what we're interested in, of course, we're here because we're neuroscientists and we want to understand the brain. Because if we can understand the influences that change how our brains develop, how our kids' brains develop, and the things that we're at risk for, we can design better interventions and therapeutics. And we can figure out who's really at risk and how to intervene. But I would argue, in order to really understand even your brain, your child's brain, your grandchildren's brain, future generations' brain, we need to go all the way back to trying to understand what happens and what is the signals that are brought together at fertilization. So within my lab, the research that we do, because we're interested in transmitting these stressors across generations, we think a lot about this single cell here called the egg. It's the largest cell in your body. It contains more mitochondria than any other cell in the body, and it is incredibly interesting in terms of the experiences that this one egg has as it matures and develops prior to fertilization. But we're also very interested in trying to understand these little guys here, the sperm. These are all germ cells. They mature at different rates in men and women, and they're exposed to different perturbations in the environment and have different lasting effects. So I guess what I'm saying, in order for us to understand this child's developing brain as a baby, we need to think way back. We need to understand mom, grandmom probably as well. We need to understand the influences and effects that mom has had during her lifetime, the impact on her reproductive tissues, and as it presents itself toward an egg, for instance, as an example, as well as understanding dad and dads, and all of these things coming together. So I always like to joke that I am a trained neuroscientist, but really I'm kind of a neuroendo-reproplacental germ cell andrologist. Because I really need to understand all of these things, and this is where it really takes a village or a community of scientists that come together, because historically, I am now in the land where many of these endocrinology, these really important critical studies were done many decades ago using sheep as the model, even though we're using a lot of mice today. So a lot of these studies were done here at the Florey. So I want to start off with first thinking about mom. We'll come to dad in a minute. But we really want to first start talking about mom. We all blame mom for many of our problems. I have a 20-year-old. He blames me for many of his issues I'm aware of. But in thinking about mom, we really need to go back to the study that I told you about and thinking about adverse childhood events that mom experienced prior to conception. And how do we think about these adverse childhood events, especially thinking about this Baltimore population and the adversity that they experienced prior to even getting pregnant? Where will those signals lie? How will they influence the point of fertilization and the development of that future offspring? And so the critical question that I always get is, right, okay, how are you going to model racial discrimination or preconception stress, adversity, ACEs, in a mouse? That's a really good question, and it's a very difficult question to think about. Well, it turns out that mice, which are not the most cognitive of all mammalian species, they're not that smart, they're very easy to stress. And so it's very easy for us to model in the lab. In fact, we do what's called a 6-week multimodal stress. This is just showing you our little mouse, female mouse here. And from the point at which her brain would be about a preschool age, up until and through the ages of 6, 8, 10-ish is the equivalent here, we're stressing this mouse. We're stressing her by doing something different every day. Mice continue to respond to changes. We do it through a lot of sensory stimulation. So this is not enrichment, but rather something very different. So they react with their stress hormones being elevated every single day for 6 weeks. And it's that micro level, those microaggressions that are pertinent to modeling racial discrimination is what we're going for in these models. So we give them 6 weeks of that stress to occur during the points at which their brain is responding to that stress. We give them 2 weeks off with no stress, so total recovery. It'd be like if you were in a very stressful job or a very stressful family situation, and then you went on vacation and you kind of thought, relaxed, forgot all about your stresses. So this is that 2 weeks of recovery. And then we breed those females compared to controlled non-stress females. And we just ask, what are the differences in their offspring? And this is just a lot of research and a lot of data. I'm going to summarize really quickly in one slide, which is basically that female that we stressed, when she had babies, remember mice have litters, we can compare her offspring, male and female, to non-stress controls. What we see is her female, but not her male offspring, just her female offspring. Despite the fact that they're raised in they grow up in the same uterus, they're more stress reactive. These are stress hormones. So this is all as an adult. Her offspring are adult now. They're more stress reactive. They're prone to diabetes, and they have an increased body weight, whether or not they're controlled or we stress them more as adults. So what this is saying to us is that this really important signal, a mom's adversity, those high ACEs, those adverse childhood events, pass on a signal at fertilization, even though she's not being stressed anymore, that impacts specifically her female offspring. Now think about that. If she is prone to all these things, increased stress reactivity, diabetes, and increased weight gain, what happens now when she gets pregnant and her future offspring? And you can imagine a cycle where this phenotype is perpetuated generation after generation of living in the city of Baltimore with discrimination that is present and health disparities, that you could end up with a lot of the issues that we're facing in the city today. But what about their brains? So I'm going to show you a comparison. I'm going to show you a heat map. And this is really a way in which we can take these offspring, they're now adults. I'm going to show you the male brain versus the female brain. What I'm showing you based on the color is just showing you all the genes. If I take out that female brain and that male brain, mice, not humans, and we do what's called an RNA sequencing, which just means we're asking everything, every gene that's being expressed in the brain, and we look at its levels compared to a non-stressed offspring, a non-stressed comparison. What do they look like comparing the males to the females? Remember, the females are the ones that are showing the phenotype. The males are not affected. And look at the differences. It's a huge difference. I don't need to even tell you what this data mean for you to say, wow, these are all the differences that happen to the female brain. These are the differences that happen to the male brain. So again, there is a programmatic change that happened when we stressed mom that she passed on to her daughters that she did not pass on to her sons. Okay, so what about pregnancy? This is going to surprise nobody in this room that mom's experiences during pregnancy can have an enormous effect, both enriching and in terms of risk, of all the things that she might be exposed to. Those can be things like virus exposure, these can be things like obesity or dietary challenges, and these can be things such as mental health issues, stress, anxiety, and depression. All of these factors, there's plenty of evidence, a lot of research being done here at the Florey that supports the fact we need to understand how these influences impact male and female offspring and why it's so important. But what you may not appreciate is that when it considers stress or impacts during pregnancy, it is actually boys who are at the greatest risk. Females exposed to perturbations while they're gestating, right? So boy baby versus a girl baby. Anybody here work in a NICU or as a neonatologist or work in the medical field, they will oftentimes tell you that boys end up in the NICU much more than girls, that girls go home before boys, and that perturbations that happen during pregnancy are much more likely to affect a boy baby than a girl baby. There seems to be a biological way in which, and we think it's the placenta, and I'll show you in a second why we think that, puts on a brake for females and it doesn't for males. And so there's a way in which the environment is affecting brain development of boys much more than it is for girls, which lends itself toward understanding neurodevelopmental disorders. If you think about ratio of diseases that are neurodevelopmental in origin and compare sex for presentation, autism, schizophrenia, ADHD, what do you know? Way more boys than girls. There's something about the intrauterine environment for when those sorts of events happen that affect the boy's developing brain that the girls seem to be protected from. Now, once we're born, that flips, and girls become much more vulnerable than boys. Okay, so there's a gene that we determined, that we isolated years ago, called OGT. You don't need to understand what it does, but this gene happens to be on the X chromosome. So we've sort of termed it the queen. It is an epigenetic regulator. It is one specific gene. It is on the X chromosome, and it escapes X inactivation. You don't need to understand what that means, except for you know that girls have 2 X's and boys only have 1, right? Boys have X and a Y. So it happens to be that in the placenta, because the placenta is the same sex as the fetus. So if you're a boy baby, the placenta is male. And if you're a girl baby, the placenta is female, based on its sex of the cells. And so in the female placenta, which is XX, they have way more of this epigenetic regulator, which is why we're calling it the queen and not the king. And in fact, this epigenetic regulator sort of determines everything about how the environment, mom's environment, transmits information to the fetus. And if you have more of this, you're protected. And so the females seem to be protected. And if we manipulate this one gene and we can genetically reduce it, we can actually make the females more vulnerable. And in fact, I'm showing you here years and years of work in one specific graph. And you don't need to understand it, except for I'm comparing the male and female placenta just to demonstrate for you. All you need to know is that a red dot, this is looking at everything, every gene expressed in the placenta. And this is true for human placenta as well, not just mouse. If I compare the differences here, a red dot just means it's statistically significantly different, meaning there's lots of differences between a male and a female placenta. Okay, so that might be part of the reason why they're more vulnerable in terms of passing on information to the brain, right? There's differences here. Now, if I take that OGT gene, which is X-linked, and I genetically reduce it, so now in the female, because she's XX, I'm genetically reducing it, so now her levels are the exact same as a male's, right? So even though she's XX, her levels of this one gene are now in the placenta the same as the male's. What happens to all of these red differences? They almost completely disappear. So now by manipulating this one gene, I can make a female placenta behave a lot like a male placenta. Okay, that's interesting. It's why we do mice, because we can play with their genes. But what does that mean for the brain? Well, let me tell you what that means. This is a heat map, kind of like what I showed you earlier. This is a female brain during development. This is a male brain during development. All you need to understand is that each column here is a different animal. Each row is a different gene, right? So they're clustered here. These are highly expressed genes. These are lowly expressed genes in the female brain. In the male, it's the opposite. Those same genes are lowly expressed, and these same genes are highly expressed. We know a lot about the brain during development. There are big differences. That's not a surprise. But what is a surprise is if I go back now and manipulate that one gene again so that the female placenta is kind of like a male placenta, and now ask about the brain, who does it look more like? It looks like a male brain. And so what that tells us as researchers is that we've identified one gene in the placenta that we now know to be really important for all of the endocrine, all of the signals that are going to that fetus during development that may be relaying information about the environment. Mom has a virus, mom is stressed, mom has a dietary challenge, mom's diabetic. All of that information interacts with the placenta and feeds its information to the brain. And so again, males and females are different. We know that about their brains. And so now what we're saying is that there's a gene in the placenta that we now know is critical for this. And we can now move this translationally. We can start to begin to identify in humans, which is where this work has gone, the same gene is regulated in the same way in a human placenta. Okay, what else can mom do? Well, there's another way. In the microbiome, I know if you pay attention to any of the scientific news to the lay press, the microbiome is incredibly important. We all know this. But what you may not appreciate is that when you first, your gut, right before you're born, your gut is completely void of any bacteria. The way that you pick up your microbiome, your first inoculant that your gut sees is traveling through the birth canal. Mom's vagina, we can all say that because we're adults here, mom's vagina is full of bacteria. And evolutionarily, this was a way in which babies passing through that birth canal get that first inoculant. And that inoculant, we've shown over the last decade or so, is really important for educating the gut. This is really important. We know that C-section babies are at much greater risk for all kinds of allergies, have differences in how their brain responds to the environment. We've done these studies in both rodents and translationally in humans. We know this to be important. Okay. So you've read all about the microbiome. You know that your gut microbiome is really important, how it responds. You know that developmentally we need to be exposed to germs. There's the whole cleanliness hypothesis, right? We know that if you're in the hospital and you're exposed to a pathogenic strain, all of these things we know to be important. Okay. So what you may not appreciate is the microbiome is even bigger than that. That we know that the microbiome of a cow, for instance, contributes greatly to global warming. And in fact, livestock, including buffalo and especially beef, and within the US produces 1/4 of our methane production, contributing to climate change. And this is really important. If you look over, 265 pounds are produced by all of the cattle around the world, largely being produced by, yes, their farting and their burping is really important in the contributions of methane. Okay. So once again, Australia has come to the rescue. So it turns out that kangaroos, eating virtually the same diet as cattle do, produce very little methane, it was thought. So these studies were done back in the 70s and 80s. And the fact that they thought that kangaroos eating the same diet, producing very little methane, could actually save the world and the planet by doing fecal transplants. This was actually an idea that was dreamed up by scientists here in Australia. They thought that if they actually took the microbiome from a kangaroo and we transplanted it into cattle, we could actually significantly reduce the amount of methane produced by this, despite some particular concerns, of course, in terms of how this might contribute to changes in kangaroo and or the cows. So I have bad news to report, though. In fact, recently, scientists actually put together kangaroo. They put 10 kangaroo into a sealed-off room, and they fed them, and they listened and measured their methane production. And while kangaroo apparently don't burp very much, they do in fact fart. And in fact, their methane production is higher than first thought, and so we will not be doing fecal transplants to save the world from itself and our meat production. So yet another day goes by where these beautiful kangaroo cannot save us from ourselves. Okay, so going back to science. So the importance of that first inoculant, as I said, this is actually a mouse pup that has been delivered in our lab by cesarean section. We can inoculate with whatever we're going to ask the question about if we think it's important. For instance, if mom has been traumatized or stressed, does that change her vaginal microbiome, and does that have any impact on how this brain develops? These are studies that are ongoing in my lab. And as I told you before, it's really critically important because your first inoculant in that sterile environment is at a time when your brain is dramatically developing. And the nutrients and that education that goes on, because this yellow lining of your gut is called the mucosal lining, it protects you from septic shock from the microbiome in your gut, has not developed yet. And when you first get that inoculant, the cells that line your gut are being educated along with your immune system. And the education goes like this. Hey, we're going to hang out here in the gut. And the immune system says, okay, as long as you stay there, we're fine. And we're going to gather some information as to who's going to be allowed to stay at this party. Right? It's who gets invited and who gets to stay, is largely how the microbiome educates the gut and the immune system as you continue to develop, and as the brain continues to develop, shaping and guiding the changes that happen in this immune system all the way through and in aging is essentially important. And for those in the audience who consider themselves senior citizens, where we start to develop more of a monotonic diet, this is the important message of the evening. If you walk away with nothing else, it is not the fat in your diet you need to worry about. It is the fiber. Eat all the fat you want, just get some fiber. So these studies have been done. We actually just published a big study on this recently. The tremendous contributions and changes to your gut related to your health, your brain health, your physiology, it's all about the fiber in your diet. So eat whatever you want, just get lots of fiber. Okay, so the vagina, back to the vagina. The vagina we know to be very important. There are current studies going on around the world now to take C-section delivered babies and actually find ways to inoculate them from mom's vagina. This is both very important because if you don't get that inoculant from mom's vagina, where do you think you're going to get it when you're a C-section delivered baby in a hospital? You're going to get it from the hospital, and that's also not good. So they're coming up with ways to figure out safe inoculation happening in C-section delivered babies. Stay tuned for those results. But what I want to quickly tell you about are some studies that we're doing, developing what we're calling humanized mouse models. Back to those traumatized women, especially in high ACEs in the city of Baltimore. And these studies were actually done by Jacques Ravel and Rebecca Broughton at the University of Maryland, where they actually characterized the vaginal microbiome of women in the city. And what is most common, so the majority of women in this room, if you actually look at their microbiome of the vagina, unlike the gut, diversity is good in the gut, diversity is bad in your vagina. So what you see here is the red color, and you see menses happen that change it slightly and it goes back. You want to have not a lot of diversity, as you can see happening here. So this is called community state type 1. This is the normal. What happens in a lot of women who've experienced a lot of trauma or adversity in their lives is they develop what's called community state type 4. And you can see all of these colors here. And in fact, this very healthy red that's showing here almost never shows up there. And these women are very prone to all kinds of infections, and they're at great risk for communicable and sexually transmitted diseases as well. So we wanted to understand this different. Is this also contributing? If babies passing through this birth canal, are they at greater risk? Are they getting a different signal than babies passing through here? And the way that we've started doing that is we can grow up these cultures. We can then take C-section delivered mice right before they would normally be born, so they're not passing through the birth canal. We can inoculate them with that little pic that I showed you. We can transfer them to a surrogate mom. We can have both control and those inoculated with the CST4, so they're growing up in the same environment. And we can now ask, how different are these animals? Well, it turns out they're already pretty different. These are brand new studies, and I'm just going to show you, if you can make that out, it's a little bit fuzzy here, but these are offspring as they've grown in that environment. They're almost fully adult now. Those with CST4, both CST4 inoculation versus normal or control, you can already see males and females already, as they hit puberty and into adulthood, they're already gaining much more weight, which may be a risk factor, of course, in our environment. And if you just look again at their brain, here's the CST1, again, the genes compared to a control brain. And you can see where it's yellow, it's mostly yellow. Where it's mostly blue. The CST4 is quite the opposite. So their brains, now what this might suggest is that it's a signal that's giving a different rate of development. This may not be disease, but what it might be is shaping the rate at which we develop. And that could make us at greater risk for other disorders. Okay, so lastly, I want to quickly talk about dad. It's really easy to blame mom, but I want you to leave this talk thinking it's not just mom's fault, it could also be dad as well. And all of the traumatic events that dad has been exposed to across his lifespan, and we know this from these studies that happened during the Dutch hunger winter and the Swedish famines. Many, many epidemiological studies have come out of both of these traumatic experiences. Things from the Dutch hunger winter, including smaller birth weights that happened across 3 generations, increased risk for different cardiovascular disease, as well as increased risk for schizophrenia in boys. We also know from this Överkalix region of Sweden where the famines have happened, and they kept incredible church records. If you've not read these studies, they're incredibly interesting. And they've looked at when different exposures and experiences happened. And this really was our first evidence that it wasn't just girls, it wasn't just mom's eggs that could transfer information, that actually boys who experienced famine during particular windows of development could pass on effects for multiple generations related to longevity. So if you had been exposed, if you're a boy between the years of 8 and 10, and you experienced during those years of a famine year because of being so isolated, that famine itself produced in your children and grandchildren greater longevity of life, which is super interesting to think about those metabolic signals that must be affecting those germ cells passed on. And this could be anywhere from 10 to 30 years difference in lifespan. So super interesting, but it was the first evidence that we had in science that really made us start thinking, what is it about the environment that is impacting dad's germ cell and not just mom's environment? Okay. So across Europe and the US, I'm not sure it's such a fascination here in Australia, but in the US we're fascinated by our dropping sperm counts. And here's all the statistics showing you this. What is causing stress in our environment? Is it endocrine disruptors? What is changing our sperm count? What is the trauma? What is the adversity? I have outgoing hypotheses. You may agree or not agree. I think the sperm counts have dropped dramatically in the last 3+ years. If we can only get him out of office, I predict those sperm counts will rise back up. So what is it and how do we model dad's experiences in our animal models so we can also translate them back to humans? So the one thing I like to point out is the difficulty in studying a female. So females, if you're going to try to understand mom's environment, what she was exposed to across her lifespan, what happened during her puberty, etc., she's the one carrying the baby. So how do we understand her germ cell when she's the one incubating the child? She's also the one nursing the child, so it's also really hard. And in the world of rodents, especially mice, same thing happens. Mom carries the litter, mom nurses the litter. Dad is really only around for very limited aspects of it. So trying to understand what mom is exposed to, things like her uterine environment, the pregnancy itself, the parturition experience, which can be traumatic, her lactation, as well, of course, her behavior. But dad, we got one cell. In the rodent world, dad is not required. And so we can very easily put dad into a cage with his females for one really glorious evening and then take him out. And so he doesn't interact with mom throughout her pregnancy. He doesn't interact with mom after she gives birth. Their children never see dad. And so we can really, in a very scientific way, we can isolate exposures. Plus, it's really easy in mice and in humans to get access to these cells if we want to ask a question. Pay them well, and you can gather cells. Okay, so how are such effects passed on? So this is where the field, and I know Tony has done, and others at the Florey as well, has done a lot of these studies, so I don't want to take away from that at all. One of the difficulties in trying to understand dad's germ cells is that dad is constantly turning over his sperm. So if that's the case, how do we ever attribute when a traumatic event happened with a signal being taken by that sperm to the egg? And this was really stymied the field for a long time because sperm are transcriptionally inert. And what that means is, as a cell, here, the sperm, as a cell, they are not capable of interacting with an environment. Something's happening, you're very stressed out, whatever is going on, you have a virus. They're not capable of incorporating that signal into the sperm itself. And in fact, after it leaves the testes and as it's transiting in, we'll come back to this in a second, what is it interacting with is the question that this field has really jumped on. How does dad's sperm, how does that germ cell interact with its environment? And this has really led to where the field is right now in trying to understand, as sperm leave the testes, after they undergo spermatogenesis, you now have a fully formed sperm, but it's not mature. When it leaves the testes, it's actually pushed out by a fluid motion, and it's not mature, it cannot fertilize, and it cannot swim yet. It's pushed into this top part of the epididymis here, which are these tubules. This is a cross-section of one of those tubules. These orange cells line those tubules, and these orange cells, just showing you here, are secreting all kinds of factors that are necessary for that sperm to gain maturation state. It is here where those sperm may interact with the environment, and these are called somatic cells, meaning they're not germ cells themselves, but they're part of the reproductive process. And if those cells store a memory of all the adversity you have experienced across your life, you can now imagine a way in which these cells can respond to the environment and produce different signals that interact with the sperm. Okay. So one of the ways we can model this in the lab is we call stress in a dish, because we want to get at the really specific signal of these cells. These cells are called epididymal epithelial cells. We can culture them, we can give them stress hormones, and then we can actually collect what's being secreted by these cells, and we can ask questions about it. We can say, are they different? Do they carry different signals? Do they have different proteins? And we can try to understand. But the coolest thing that we can do in the world of mice is we can take those vesicles, they're called extracellular vesicles. Here are some from a non-stress dish. Here are some from cort, which is the stress hormone, cortisol in humans, corticosterone in mice, right? So we've taken them from stress experience, okay? We're now going to take a male mice, and he's going to donate sperm for us. We're going to divide it into 2 pools. Same dad, 2 pools of sperm. These we're going to incubate with the controls. These sperm from him we're going to incubate with his previously stress exposed. Okay, everyone with me? And then we're going to take eggs from the same mom. So the same female is going to give us a bunch of eggs. We're going to perform a fertilization technique called ICSI, intracytoplasmic sperm injection. We do it in humans as well. And we can say we can do some of the eggs with these sperm, and we can do some of the eggs with these sperm. And we can actually put them into the same donor mom, left side of the uterus or right side of the uterus. So now we have same dad sperm, same mom egg, same intrauterine environment. And we can then harvest those embryos during development and say, aha, is there anything different about how these guys are developing? Is there different with their brain, most importantly? And we also can do follow-up experiments. We can put them in different ones and let them grow up and develop into healthy adults and ask, are they different? And this is a really amazing graduate student's thesis work, 5 years, 1 slide, is what I'm telling you here. And you don't need to understand anything except for the answer is, oh my gosh, midway through gestation, their brains are dramatically different already. And not only that, but if you look at what's different, there's things called synaptic signaling, cell-cell adhesion, transport. There are really vital signals that are being changed just from what we incubated that sperm with. And what these signals are probably telling the developing embryo is, it's a stressful environment, you better hurry up and develop faster. And we see this in both humans and in rodents, that this stress signal, which we think is being transmitted by these vesicles, is really producing that faster rate of development, which is leading to these differences. And just lastly, this here, this is actually looking at, this is your stress response. So everyone in this room has the same stress response as a mouse does. If I were to acutely stress you right now, let's say pass out an exam and see what you've learned tonight, everyone getting a little bit stressed? Or have you come up here and give a talk about what I just talked about? And I was to look at your hormone response. Here's your starting point. Here's how high your stress hormones go, and then 2 hours from now, how they recover, okay? What this says is that the animals that grow up as adults that came from the sperm, from the stressed vesicles, have a much higher stress response. That's what this data is telling us. So we know that not only is it shaping the brain, is it speeding up the brain developmental rate, but it ultimately leads to an animal who is more less stress reactive. I was thinking of our female studies. These guys are less stress reactive. So what that says is probably dad's way of taking cues from his environment, evolutionarily, has been preserved, that he can send signals. It's not predicting a disease state, but what it's shaping is how that brain is developing for a best fit for its environment, making it less stress reactive, which is important. If you're going to be in a chronically stressed environment, it behooves you to not be dramatically stressed all the time. And that's what we're seeing in these animals. So I'm going to end with, what does this mean for human beings? And as I said, all of our mouse work in my lab also has a human component. This is one of the easier ones to do. We actually did this when I was a professor at the University of Pennsylvania in Philadelphia. We did this in collaboration with a psychiatrist, Neil Epperson, in which we wanted to use the stress of undergraduates to study this question. And it turns out, if you pay them enough, they will come into the office, not my office, not my office. They will come into the fertility clinic where there's lots of magazines and privacy, and they will donate every month over the course of 6 months. And during that 6 months, they're also undergoing final exams. And so we can track with the psychiatry department their perceived stress state. We can follow that stress state as well. And we can also, because not everybody's going to be stressed. I mean, anybody have a kid who's at university and doesn't really particularly be stressed about much, right? We have those kids too. And so we follow their stress state, and we follow the collection of their sperm. And these studies have never been done before, believe it or not. We know so little about human sperm and the dynamic processes to the environment. And so we collected across 20 different individuals. Every month they came in, they went to psychiatry, filled out their perceived stress. They went over to the fertility clinic, did their business, got paid, and we followed them over the course of 6 months. Okay, so what this is, you've seen so many heat maps now, you know exactly what I'm showing you, right? So what you're seeing across here is, again, every column is one of our 20 individuals and one of his 6 months. So there should be a, give or take, 120 samples. Sometimes one of the guys didn't show up. And we sequenced all of the RNA in their sperm. These are the signals we're talking about, okay? And so each row is a different RNA. So each row is an RNA. There's thousands of them in human sperm, just like mice. And each column is a different individual. The color on the top here is saying that when we do a hierarchical clustering of what's there and how much of it's there, most men, you'll be glad to know men, you sort of cluster with yourself. That's good. That means from month to month, you're not that different from the previous month. Okay, so that's what that means. Now, can anyone, even in the back, tell me, do you see two distinct patterns of genes that are, based on the color, more highly expressed and more lowly expressed, and more lowly expressed and more highly expressed, right? If I were to put a line down the center here, yeah, you see that? Well, it turns out when we do that and we looked at the stress responses of these individuals, the no stress guys were on the left and the stress guys were on the right. And so that tells us that we're on to something, that there is something about signals and the perceived stress, and that resolution from that stress is what we're looking at now, and how those signals may change. Now, obviously, we then did not pay the undergrads to go and have babies, so we don't know what these signals mean, right? We don't know that yet. But what we can do is we can model this, and so we're working with the Genome Sciences Institute at Maryland, and we were able to take all of their stress and all of their sequence, and we actually came out with a couple of RNAs, 2 or 3 of them, which was truly amazing because this is only 20 guys, and humans are incredibly complex individuals. We came up with 2 or 3 that really fit our statistical model, and when we look to see what those RNAs are, it turns out they're involved in things like fertility and the rates of development. So when it comes back around again, the stuff that we figured out with the mice seems to be coming true also in the humans. And that may fit again. If it's a stressful environment and we're not all going to survive, well, we better be out there breeding, right? And the rate of development better be increased. And that's sort of what we're seeing. So things that we all kind of know in the back of our minds, we're actually figuring out some biology behind how this happens. So in the end, why does your brain, as you sit here today, function the way it does? Well, it's complicated. And I think the more that we learn, the less that we really understand. And whether or not, there we go. And all those things are actually, the more we learn, the more things seem to be going up in smoke. But I do think that we're making progress in trying to understand this interaction between dad and mom that gives rise to differences in our offspring, our grand offspring, and maybe even our great-grand offspring. But the thing that you can go home with tonight, if you don't have enough at cocktail parties to talk about between sperm and placenta, is that it's pretty clear we can probably still blame our parents for most of our problems. So thank you, and with that, I really need to make sure I have incredible talent in the lab. All the individuals, these are most of the current members in the lab that have contributed to all of this work. As I said, Jennifer Chan, who's actually not even in this picture because she's off doing her postdoc at Mount Sinai in New York. And I wanted to highlight that our funding in the US comes from the National Institute of Health. And the studies, the last ones I talked about on sperm, believe it or not, are actually funded by the National Institute of Mental Health. And so I really am thankful to them for buying into the fact that we need to understand, and they allow me to write things like sperm into my grants to NIH. And of course, you can always follow us on Twitter, and please do. And I'm happy to take any questions.